Okay, looks like I'm live. Hello, and welcome back to our Edvotek Biotechnology Education live streams. My name is Dr. Danielle Snowflack. Those of you who have joined us here before will have met me. I am the Senior Director of Education at Edvotek, and I'm so happy to be here again for our first biotech workshop of the new year, where we are going to be talking about protein electrophoresis, which is a technique that I love and I've used a lot in my scientific experience. And I hope that, through this workshop, you'll learn about this technique and you'll want to bring it into your classrooms too. It's a little different. We generally talk about DNA. Today we're talking about proteins, but I think that it'll be a really fun addition to your biotech lab. For those of you who might be joining us for the first time, Edvotek is the biotechnology education company. We were founded over 30 years ago by Dr. Jack Trickshian, who was a professor of biochemistry at Georgetown University. This was a really exciting time to be in the biotechnology lab. I always like to point to, what was happening in the 80s, like with the isolation and creation of recombinant insulin protein to be able to treat diabetes, or the use of DNA fingerprinting to, identify people, or PCR, the polymerase chain reaction, which we hear about a lot recently, which is kind of like a DNA photocopier that we can use to detect diseases in people. And so these are all really amazing innovations, but very few of these were being translated into the teaching laboratory for students. And thus, Edvotek was born. And today, while our, experience is in education, we work with educators, with biohacker spaces, with research labs all over the country to foster the next generation of scientists through hands-on active learning activities. So thanks again for joining us today. Let's talk about protein. So protein is going to be everywhere. And so I feel like especially this time of year, we're all focusing on the food we're eating with our New Year's resolutions to be more healthy. And so we talk a lot about protein in the context of food. What food is high in protein? Am I eating enough protein? What does protein even do for me? Is it good protein? But you know what? None of these questions actually matter when it comes to what's happening in our cells. From a cellular perspective, proteins are the workhorse molecules in our bodies that are really responsible for the day-to-day function of our cells. They allow our bodies and our cells to function properly, because they perform so many important functions within the cells. And so I listed a couple of functions on this slide over here. The directions always get me messed up. But yeah, so we're looking at this slide. And so some of the important functions that proteins perform are in enzyme catalysis, where they can increase the rate of chemical reactions that are happening within our cells. Proteins are important for maintaining homeostasis in that they help an organism stay alive by reacting to different environmental conditions and stressors, and help us maintain, optimal performance. Proteins bind to and transport small molecules throughout the body through cell membranes and even within a cell. Proteins interact with DNA to turn genes on and off, and this is called gene regulation, and it's really important. We also hear a lot about the immune system and antibodies. Antibodies are specialized proteins that the body uses to identify and attack pathogens, keeping us healthy, or at least helping us to prevent severe disease. And proteins are important to give a cell structure. Proteins are found in plant and microbial cell walls to provide structure. They're present in cell membranes to be able to anchor cells together to form tissues. And they're even found inside the cells to give structure to the cell as part of the cytoskeleton. And so these are just a few functions of proteins. And I think you'll agree that they're pretty fantastic molecules, and we're all really grateful to be full of protein. And so today, we're going to be talking more in depth about proteins as we do a protein electrophoresis experiment. Proteins are polymers. They are built from individual amino acid building blocks. The amino acid sequence of a protein determines its size, its shape, and its charge. And so there are many different biotechnology techniques that we can use to tell us more about proteins, including mass spectrometry, Western blotting, chromatography, and more. In this workshop, we are going to be talking about Sds Page, which is sodium dodecyl sulfate polyacrylamide gel electrophoresis. And this is a technique that we can use to find the molecular weight of proteins. We're going to be performing a basic electrophoresis experiment, which is the determination of protein molecular weight, Kit 153, which is an Edvotek experiment. This demonstration will be recorded and our slides will be available on the website. If you'd like to be notified when they're posted, please fill out the form that Maria is going to put in the chat box. This form will allow us to get your email address and then contact you to send you the slides. We're also going to be offering professional development certificates to those who are watching live. So if you want us to send you a certificate, please be sure to check the box on the form. The link will only be available for 1 hour after the presentation, so be sure to fill it out before 4. So let me actually grab that Google form link for you. And this should, oh, there, now it is. Now I see it. Thank you, Maria, for posting the form. So yeah, fill out that form and let us know. Also, there's a comment box on that form. If there's any other biotech workshops you'd like to see, definitely let us know because we will, we're always looking for new ideas and, new workshops to put on for y'all. Okay, so let's get into the experiment. So this experiment's going to take a little longer, so we are going to load the gel as quickly as possible, and then we are going to talk about what's happening while the gel is running. Just to give you a quick overview of electrophoresis, we are going to be using electrophoresis, protein electrophoresis in this case, to analyze the size of unknown proteins. Sds Page is a technology that uses a porous gel matrix and an electrical current to separate protein molecules by size. It's a little bit more complicated than that based on the chemical properties of proteins and how they interact with one another, but we'll get into the ways that we address that problem later. This slide is an overview of our workflow for an electrophoresis experiment. We are going to submerge a precast gel into the buffer and then load our prepared samples. We attach the safety cover with the leads and then turn on the power supply. After about 30 minutes or so, we can check on the results of our experiment. And in our case, we're going to see our protein samples moving through the gel. So in preparing for protein electrophoresis or any electrophoresis, one of the important preparation steps is making the gels. And so in our case, let me switch my camera view to go to the webcam view. I'm going to put some gloves on. Again, we always want to make sure to use the proper personal protective equipment when performing experiments just to keep ourselves safe. So I am going to wear gloves. You're going to want to wear eye protection in the labs. Again, everything we're doing is pretty safe. But it's always best to be careful. And one of the reasons we use precast gels in this experiment in particular is because of the polyacrylamide matrix. Now the polyacrylamide matrix, and so let me actually take out one of these precast gels so you can see it. And so all electrophoresis basically uses electricity to move molecules through a porous matrix. For polygel, and I just removed this gel from this plastic bag. These are precast gels. And so each of these gels contains prepolymerized polyacrylamide. And this is important for us. So polyacrylamide is our matrix for separation. I kind of think of it like a science jello. This is going to feel a little different than an agarose gel, but it's still the same idea. The polyacrylamide is going to act like a strainer or sieve, which is going to allow us to separate our molecules by size. Polyacrylamide gel is a very versatile matrix for separation, and we can actually use it to separate a variety of different molecules. Just like agarose gel electrophoresis, polyacrylamide gels can be used to separate nucleic acids, including DNA and RNA. In general, though, you're only going to use these polyacrylamide gels if your nucleic acid fragments are really small. Agarose is just way easier to use in the lab, and it's way easier for larger gel fragments. However, polyacrylamide is the matrix of choice for protein electrophoresis because it allows us to get really nice bands. Polyacrylamide is a polymer gel made up of chemically cross-linked acrylamide monomers. This is going to create that porous molecular sieve. However, we don't recommend casting your own polyacrylamide gels in your classroom lab unless you have advanced students, because the acrylamide molecules are highly toxic when they're free. However, once they're chemically cross-linked, it's really safe to use in the classroom. There is a question in the chat. Should the gels come up to room temperature before running the experiment, or can they go straight from the fridge to the chamber? I haven't noticed a big difference going straight from the fridge to the chamber. I usually do that. I think that you want to make sure your buffer is cool. You don't want to use hot buffer. That would be if you're trying to run one gel right after another in the same chamber, because the actual heat of the gel there, the heat of the buffer can give you a little bit wonky results with your bands. But I have never found a difference in the gels, whether or not they're cold or not. I actually just pulled this out of the fridge before this workshop. I just happen to have it here because the fridge is in the other room. So I would say that the temperature is not going to make a huge difference. And so to use these precast gels, you are going to store them in the refrigerator because they are perishable. You're going to want to follow the directions, basically, to know how long you can keep them. But you're going to remove the gel from packaging. These are our Edvotek precast gels. Some precast gels are a little bit different, like the ones you see in the picture here, where they have a little bit of tape on the bottom. Basically, we want the current to be able to flow through the gel. But given the nature of the gel itself, when we pour into these cassettes, you need to block off the bottom. And so some gels will have that tape that they use to block off the bottom before they pour the gel in there. So you want to make sure if there is tape that you remove the tape so that the current can flow through the gel. Here we have the comb. This comb is going to cast the wells in our gel. That's where we're going to put our samples. And then we remove that comb and you can see that we have these individual wells in our gel. That's where our samples are going to load. And so we then place our comb, our gel into our chamber. You're going to want to make sure the clip is facing you. And then when you put the gel in the chamber, the high plate is going to go towards you, at least in the Edvotek chambers. And that just is because of the way the current is going to flow through the gel, in through the top and then down through the bottom. We then insert the gel into the tank and we're going to submerge it in buffer. All right. So we're going to load these gels right away. So what do we need to do these electrophoresis experiments? So first, we have our rig, we have our gel, which I told you about in the last slide. We do need our samples. So we are going to be using Sds Page to analyze 3 unknown proteins, and we're going to have a ladder as well. The proteins for our kit come in the lipoprotein format. I actually ran one of these gels last night because I wanted to see how it turned out. I wanted to check out the results and make sure everything looked good, and I wanted to prepare a pre-stained gel for you so I could show you. But basically, the proteins are going to come freeze-dried in our lipoprotein format, and that makes the samples more stable for storage and for shipping. And it helps you get the best quality proteins for the experiment. The samples also contain the loading buffer, which is going to make it more dense than water, which is important because we're going to be loading samples into wells that are under buffer, and we want to make sure that the samples stay in the wells. And this is also going to contain special chemicals that allow us to analyze the protein. And I'll tell you more about that as we discuss the nitty-gritty of proteins and electrophoresis. We are going to submerge this gel in buffer. So the buffer is going to be a mixture of salts and buffers and detergent. And there's a couple of reasons why we need to use buffer instead of water. The first is that electrophoresis uses electricity to separate molecules. And water itself alone is actually a very poor conductor of electricity. And so we need to make sure that there are molecules that can conduct that charge. And that is what we do find in this buffer. Next, we are working with biological molecules, and we want to make sure they are at a charge that works for the protein so that it remains charged and it can be separated by electrophoresis. Next, we actually have a detergent, and we need to keep the proteins at a negative a negative charge to be able to be separated. And third, we actually have detergent in this buffer, which helps keep the proteins denatured. So the buffer comes to you as a 10x concentrate here, 10x trisglycine buffer, and then you would dilute this in your lab. Next, we're going to get the samples into the gel. I am using an adjustable volume micropipette, a standard micropipette you'd use for most of your experiments. Let me aim this down a little bit so we can see the gel chamber fully. You can use any kind of tip to load it, either a regular yellow tip or a gel loading tip. The gel loading tips are going to make it easier, but I'm going to show you how I do both. So micropipettes are going to be is a precision instrument that allows us to load samples, small volumes of sample and measure them accurately. This first sample is our component A, our protein marker, and I'm going to load 20 microliters of it. If you want to learn more about pipetting, we have a workshop that focuses just on that. And so with these gels are a little trickier to load than your agarose gels, just because the wells can be a little more difficult to see. And so you really want to kind of get your eyes down, kind of level with the gel, and what you should be able to see are the individual wells of the gel. And they don't really pick up well on the camera. You can maybe see a little bit there. I like to take my pipette tip, put it on the taller plate, and slide it down until my tip is just above one of those wells. And then I slowly and evenly push the plunger down so that the liquid goes into the well. And you can see that well has filled up with blue. One thing that sometimes people will do to make it easier to load gels, let's see, that was just sample A. I'm going to put that there. One thing that people sometimes do to make it easier to load the wells is they take the practice loading buffer. And so this is just a dye solution. It comes with the kits. And what you can do is, gently, now I'm going to put it in some of these back wells. You can just gently put a little bit of this buffer in the wells so that it falls in and it starts to label those wells. And you don't want to use too much because, it will fill up your wells with stuff. But you can use just a little bit to make those wells more obvious to see. And so it's very subtle on camera, but you should be able to see that these wells here have a lighter blue color, and that just allows us to see them. I'm going to load sample B. Again, this is component B. This is our pre-stained protein 1, our protein of unknown size. Again, I'm going to load this into here. Gently, again, sliding down, loading the protein into the well. So that's how I do it with the yellow tips. The nice thing about the gel loading tips is that they have a finer point, and it's going to make it a little bit easier to to get the protein in the well, especially for beginners. The one caveat is that they are kind of sharp, and so you do need to be careful that you do not puncture the bottom of your gel. It's not going to be as big of a problem as it is with agarose electrophoresis because the samples aren't going to fall out of the well, but it'll definitely affect how nice your bands are. All right, so let me put this here. And so again, you're going to take the fine point, you're going to just slide it into the well. And then you can see the sample just being loaded into the well. And so this is tricky. It does require practice. I've loaded a lot of Page gels over my career, and so I'm pretty comfortable doing it. But it is a skill. Any of these techniques are definitely a skill in that, they take practice to run. Do you think I have 4 samples in here. Let's run those 4 samples, and then we'll see how they turn out. So let's see what else. Okay, so we're going to put the chamber on here, the lid on the chamber. So this chamber lid contains our electrodes. These are wired so that you can always put it on in the correct direction. And it's wired and color coded to help you put it on in the correct direction. You want to have the red side and the black side in the right place. These chambers have these large push buttons which help you get the lid on and off really easily. And then the edge is rubberized because sometimes with a lot of buffer being around, your hands are going to get wet, and it just gives you a really nice grip on here. So we plug the leads into our power supply. I'm using the Duo source today. It is behind this chamber. I'll show you a picture of it on the next slide, but I'm going to plug it in. I'm going to turn the voltage to 150 volts, and we're going to go. So just for reference, this is one of our protein experiments. It needs to be performed in the MV10. If you do have agarose electrophoresis chambers and you want to try protein electrophoresis in them, we also do have kits where you can do protein separation with agarose if you just want to try proteins in your lab. But this is going to be the way that most labs would analyze protein samples. So when attached to a power supply, the electrodes are going to generate the current that we need to push our proteins through this gel. And so on this slide, so you can't really see the duo source, on this slide we have our 2 electrophoresis chambers. The one I'm using is the duo source 150, which can run up to 2 gel chambers at 75 or 150 volts. For protein electrophoresis though, I don't have a duo source here to use in my basement laboratory. However, I do think that if you have a quadra source around, an adjustable voltage electrophoresis power supply, I do think it's nice because you can adjust the voltage based on the percent of your gel, which can give you some nicer results. And so this unit, the quadra source is adjustable from 100 to 300 volts. And so again, you can use any power supply you have. Like the only special piece of equipment you'll need will be the MV10. But I do think the quadra source does give a few advantages when it comes to protein electrophoresis. Okay, so now we've got our experiment running. I'm going to leave the camera on the experiment so that you can see the progress of our samples as they run through the gel. And we're going to talk about proteins, like what are proteins and how do we use electrophoresis to analyze them. And, oh no, that's the right slide. Okay, so basically proteins are coded for within our genome, and they are represented they're represented in our genes, which is a known unit of heredity with a known nucleotide sequence. So most genes within our body are going to code for proteins. Again, proteins are a major component of every cell in our body and they do a lot of functions. But there are genes that are going to code for ribosomal RNAs or transfer RNAs or other important molecules. But the vast majority of genes are going to code for these workhorses in our cells. The genes are transcribed into RNA and then translated into protein by the ribosomes. And so when the ribosome is reading the DNA or the RNA, they use a triplet code in which 3 bases of RNA correspond to 1 amino acid. And there are also special codons that tell the ribosome to stop or start translation. And so here we can see there are a lot of nucleotides. Like I said, there are only 20 amino acids, but based on the code, some 3-letter codes will correspond to multiple amino acids. And so if you guys are interested in learning more about translation, we can always talk about that in another live stream. But the important thing to know is that there are 20 standard amino acids found in proteins that are coded for in our genome. Some of these amino acids are essential, meaning that the human body doesn't have the biochemical pathways to produce them. We need to get those amino acids from our food. Other amino acids are considered to be non-essential, and we can build them from the body. And so the molecular weight of a protein, which is what we are going to technically be analyzing by electrophoresis, is a function of the number of amino acids and the type of amino acids in the polypeptide chain. Again, the protein is going to be built. Proteins are molecules that are built from individual building blocks called amino acid monomers. Protein is a polymer built from amino acids. And so we form these long chains of molecules that are bound to one another that create these proteins. And a protein monomer or an amino acid, you can see a representative diagram of one here on the screen. Each amino acid consists of a carbon molecule that's bonded to 4 groups. We have an amino group, or N, a carboxyl group, or a C, a hydrogen atom, and then an R group. And an R group is a chemically unique side chain. A peptide bond is formed between the N group of one amino acid and the C group of the next one. And they're chemically linked to one another. So that's a very strong, tight, permanent bond. But because the molecules are not symmetric, you have an N-terminal and a C-terminal, we do have directionality in these molecules. And so sometimes you'll hear about something being at the N-terminus of a protein or the C-terminus of a protein. And that basically just tells you what end of the protein we're at. And the R group here. I actually have a picture of the polymer, so you can see how they're all linked together here in a long chain. And that R group that is hanging off, and you can see we have three monomers linked here to make a three amino acid polymer, and each of them have an R group. And so the R group is the chemical part of the amino acid that's going to give the amino acid in the protein its chemical characteristics. So the side chains can be polar or non-polar, acidic or basic, hydrophobic or hydrophilic. And the interactions between these side chains are going to determine a protein's structure and function. So let's take a quick look here. You can see it. It's a little bit difficult to see because of the bubbles that we have here in the electrophoresis chamber. The bubbles are basically showing us that, we have water being broken down, and that's hydrolysis that's happening at the terminals. But we can see right here are proteins, and they're starting to enter the gel where they will be separated. Okay, so protein. So let's talk about how these all work together to build to fold a protein. Okay, so proteins are going to exhibit different three-dimensional shapes and folding patterns, which are determined by their amino acid sequences. And so basically what you can, what voltage am I running out? So I am running this at 150 volts because I have a duo source at home. I believe the literature for this experiment has you running it at 125 volts. Basically that'll give you a little bit better, it'll take a little bit longer, but I think the bands resolve a little bit more nicely. And then the buffer doesn't get as hot. And so that's a caveat. The more energy you put into the buffer, the hotter it gets. And so I generally, that you can run the gels at 150 volts using the duo source. But, if I were in a lab, I'd probably run them at 125. And so, okay, so we have our proteins here, and proteins are going to they're going to create different three-dimensional shapes based on their amino acid sequences. So remember, one of the things that we say is that, chains can be polar or non-polar. They can be charged positively or negatively. And these are going to affect the way that the protein interacts with itself when it folds. So you can imagine, having a piece of ribbon, let's say like this ribbon that I happen to have sitting here. A polypeptide chain itself is just a ribbon, and this is its primary structure. But, if we tie a knot and we make a bow, we're creating turns in the protein. We're creating areas with a lot of protein residues that are folded up on one another. There's going to be pockets inside here that are not accessible, to the outside of the protein, and then say we take this and, tie it in one more knot. We're even making some of those more inaccessible as we make our, kind of globular protein. And if we think about charges, we're going to want positive and negative charges to interact with one another to hold together. We might have hydrophobic regions that are going to bind to one another, and they're going to stay in these pockets of the protein and hold it all together. And sometimes you even will get a disulfide bond, which is going to be a chemical connection that holds parts of the protein together. And so, again, we can think of, this is our primary structure. We have alpha helixes, which would kind of be like wrapping this ribbon around my finger like this. We're going to form these helix shape where we're going to get bonds and interactions between the amino acids here. We're going to get beta sheets, which would be sort of like folding this up and down on itself over and over again to form a secondary structure. And then we can even have instances where we bring, and so those are the ways we're going to form secondary structures. The tertiary structure is then going to be how all of these different structures are going to fold and interact with each other. So again, if we have a helix here, and then we have a beta sheet here. You know, how do we keep those two domains together? You know, maybe we not at all. And basically, all of the way that comes together is going to be the overall fold of the protein, which is representative of the different domains of secondary structure. Then we have quaternary structure, which is not, we don't find every protein has a quaternary structure, but basically what that would mean is that we would bring another protein in here to interact with this first protein. And those proteins would be necessary to work together for its function. And so the precise 3D configuration of a protein is critical for its function. And so, you know, I mentioned antibodies earlier. Antibodies are going to be these Y-shaped molecules. And the tip of each Y, like an antibody is going to be a Y-shaped molecule. Let me actually just draw this on the back of a paper, right? So we have these Y-shaped molecules with antigen binding sites at the end. And this structure, with the antigen binding site, it's important to maintain that structure for the function of the protein. You know, if that domain gets messed up, it won't be able to recognize the antigen, and your body won't be able to, you know, find and attack those pathogens. Or if we think about an ion channel going through the wall of our cell, if we don't have all of those proteins working together to form those channels, you know, we're not going to be able to move, you know, water or hydrogen or other molecules across our cellular membranes. And so all of these components are critically important to having a protein that can function the way it needs to in our cells. And so, you know, let's actually talk. I gave you a couple examples of proteins in this last slide. Let's talk about two of my favorites that are going to really point out a lot of important things about proteins and their sequence and how their structure, you know, or how their structure affects their function. So the first one I'm going to talk about is one that you probably use in the lab a lot. This is green fluorescent protein, or GFP. And so this GFP seems kind of crazy because it glows bright green. And, you know, it seems almost extraterrestrial, but this is actually a protein that was identified and isolated from jellyfish in the 1970s. So this isn't even a new innovation. You know, GFP has been known for a really long time. It's a small protein that's going to absorb blue light and emitting green light. And you can see, if you look at the structure, we've got some nice beta sheets where the strands are lying next to one another. And that actually forms a structure called a beta barrel. So it kind of forms this round cylinder, like this cylindrical protein with a tunnel through the middle. And the tunnel, and so these are important for its function. This structure is important for its function. In that there's a chromophore inside the tunnel. And we'll talk about more about that chromophore on the next slide. But, you know, one thing that I always like to point out is, like, once researchers were able to identify the DNA sequence of this molecule, genetic engineering techniques were used to be able to introduce this protein into other organisms like E. coli. And so if you're doing GFP transformation, you are introducing a protein into bacteria that isn't there naturally, but it can make those cells glow green. And so I mentioned the chromophore briefly on the last slide. Did the slide change? Am I lagging? Oh no, there it is. Okay, good. So basically, as I mentioned before, you know, the identity of each amino acid happening in a particular place, it's important because, you know, and it's important because when we have the structure of a protein, you know, we may have two amino acids that we need next to each other to form a particular structure. So you can imagine if we had this ribbon, if we had something here and here, they'd need to be next to each other for proper function. And that's what happens with the chromophore. So the chromophore is a special structure which is built from 3 R groups of 3 different amino acids that are brought into proximity, so brought near one another inside this beta barrel protein. And so the chromophore is, you can see the structure on this page, and that is, it's responsible for the light production. So the absorption and the absorption of blue light and the emission of green light. And so this is an awesome place to show the differences between structure and function, because if we change the amino acid sequence at that particular location, that's going to affect the pattern of light absorption and emission of the proteins. And that's a powerful tool because it actually allows us to create a rainbow of different fluorescent proteins. And I have a bunch of them here on the slide. This figure was taken from a more in-depth paper. But what you can see is that when we change those amino acids to be different amino acids, the structure of that chromophore changes and the color that we produce is different. And so that's how we can get blue fluorescent protein or cyan fluorescent protein from starting with a green fluorescent protein. And I think that's a really powerful example because it's showing that the amino acid sequence is directly affecting the sequence of the protein that you're looking at and its function. And so another thing that I always like to point out with GFP is that the discovery and development of fluorescent proteins as a biological tool was awarded the 2008 Nobel Prize in Chemistry. So not only are these like really cool experiments for showing how genotype relates to phenotype and also how protein sequence affects its function, I mean, it's also Nobel Prize winning experiments. And I think that's really powerful. The next protein I always like to talk about with structure and function is going to be hemoglobin. So hemoglobin is a more complex protein complex than GFP. So GFP is generally monomeric. It's generally one protein. Hemoglobin is a heterotetramer. And so that basically means that it is, if we break down that word, hetero is going to mean different, and tetramer means four subunits, because four is tetra and mer is a unit. And so if we say something is a polymer, it's just composed of many different units. A tetramer is built from 4 units. And so when we call it a heterotetramer, that means those 4 units are different. And so there are 4 subunits in our hemoglobin protein. The first, which is alpha hemoglobin, which is shown here in red, and beta hemoglobin, which is shown here in blue. Each monomer is going to have a heme group. So you can see the heme group is here. The heme group is important for binding and transporting oxygen. Each hemoglobin molecule has a heme group, and each heme group can transport oxygen. And so in our red blood cells, hemoglobin is going to exist as a free tetramer in the cells, and that's how we're going to be able to transport oxygen. But if we change the sequence of proteins, we can, it's going to affect the disease, it's going to affect the protein's function. And so this is evident in sickle cell disease, or SCD, which is an inherited blood disorder. This disease is caused by small changes in an individual's genome that interfere with the body's ability to create a fully functional hemoglobin protein, again, which is important for transporting oxygen from the lungs through the body through the bloodstream. When we make changes to the protein sequence, problems can occur. And so researchers have identified more than 400 mutations in the beta hemoglobin gene that can cause sickle cell anemia. And I did talk about this in my last live stream, or the second to last one. I don't even remember. But if you're interested more in the DNA changes that happen in sickle cell anemia, definitely check out my previous live stream. I'll link to it here in the video description if you want to come back to it later. But this is a genetic disease, and the most common genetic change that causes sickle cell disease is a single point mutation from A to T, which changes the amino acid sequence of the protein. This single base pair changes a single codon, which swaps out glutamic acid for valine. And these 2 amino acids have very different properties. The change from the negatively charged glutamic acid to the hydrophobic valine is going to change the protein structure. So remember, these side chains are interacting with one another to create the protein to stay in a specific order, so that the protein can then perform its function. And when we change one of those things, we change the function of the protein. And then hemoglobin can't bind oxygen anymore, and that can be problematic. Okay, so let's get back to our protein electrophoresis. Again, here's the same flow chart that you saw from before, just to kind of get you back oriented in thinking about protein electrophoresis itself. Again, so we loaded our precast gel into our chamber, added the buffer, we loaded our samples. Again, you may like to load them with the gel loading tips or the regular yellow tips. whichever way you can actually load with, transfer pipettes as well, but it's not going to be as accurate. But, you want to get those samples into the gel. We then put the lid on, turn the power on, and pass the current from the gel. So now let's actually talk about what is happening inside the gel and the kind of tricks that we have to take to be able to analyze these proteins, because the secondary structure, the tertiary structure, the quaternary structure, and the charge of the proteins, these are all going to affect how proteins move in an electrical field. And actually, if we stop right now, the proteins that I have here are pre-stained, and I'll talk a little bit about that in a minute. But if you look, you can see the proteins have separated. Let me see if I bring this closer, if it's more obvious. My camera can pick it up better. Let me see if my arm will stay down that low. I have a little camera mounted on an arm, and that is how I run the experiments. Visualize the experiments. Okay, so you can definitely see those proteins, those 3 bands separating, 1, 2, 3, and our ladder. Our ladder definitely needs a lot more separation before we can see the individual bands. Okay, so here's kind of like a representative gel. It's going to look a little different, but follow me, and we'll get through the principles of electrophoresis together. So you can imagine, we have proteins that are negative. We have protein samples that we're going to load in a gel, and we're going to pass an electrical current through the gel. However, since proteins can be positively charged or negatively charged, this is going to affect their migration. So what does this mean if we were to add proteins into the wells in the middle of this gel and apply current? Basically, we would need a gel where negatively charged molecules can move through the gel towards the positive electrode, and the positively charged molecules can move through the negative electrode. And that's because protein molecules, by virtue of their side chains and their chemical bonds, have this net charge, which affects the way they move through the gel. And furthermore, proteins are complicated. They are going to be attached to other proteins and complexes. They can have lots of different structures. So again, we could have long fibrous proteins. We could have really small, compact globular proteins. These are all going to affect the way the protein runs through the gel. You can imagine a large spherical protein will get stuck in a channel that a rod-like protein can get through more easily. And basically, because of the charge and because of the size of the molecules, we're going to separate them into discrete bands in the gel. And that'll allow us to learn something about the identity of the protein. However, this is what a Page gel looks like. And so that does not look like the gel with the 2 dimensions that I showed you. We're only running the charge through this gel in one direction. And so how do we do that? Then how do we use electricity to separate proteins using this electrical current? And so this comes down to chemistry. We need to figure out a way to make all proteins have the same charge and to get rid of some of that secondary structure and tertiary structure and quaternary structure so that we can accurately run them through the gel and to learn more about the molecular weight of the protein itself. And so proteins in their normal biologically active state are called native. And so they can be fibrous proteins, which are like longer chains that are all wound together like a collagen. They can be globular proteins like serum proteins. But these are all many proteins are very chemically stable. And so these complexes are going to affect the way the gel will be able to fit through the pores in our polyacrylamide gels. So there's a lot of things that we need to do to get rid of the net negative charge or the net positive charge. And again, those are the charge of the protein, the shape of the protein is dependent on its amino acid sequence and how the amino acids link together. That's also going to affect the charge of the protein. Protein charge then gets tricky also because if you change the pH, we can either add or take away charges from those molecules, which is going to affect the way the protein interacts with the charged gradient and moves through the electrical field. And then not only that, proteins and complexes are going to be larger than their single subunits. And so we won't be able to learn If they're all stuck together and we put it in a gel, we won't be able to learn how many subunits are in a protein, which is very important for learning about complexes. So how do we get rid of all these problems? Like, how do we take these proteins and make them uniform so that we can run them through a gel at a single current and get results? And so that is where we come into Sds Page. So Sds, again, is sodium dodecyl sulfate, and that is a detergent. And then the Page is polyacrylamide electrophoresis. And so what we need to actually do in Sds Page is disrupt the protein structure. And we do this in a couple different ways. We use heat that's going to break apart the complexes and denature proteins. We are going to, that's also going to disrupt the hydrogen bonds that happen between these individual proteins, which allows us to break them into individual components. We're going to add a reducing agent. Most times it's called dithiothreatol or DTT. That is going to break up the disulfide bonds that are chemical linkages between proteins or even within proteins, which helps us break up those structures. Heat is going to denature the protein like cooking an egg, where we go from a clear protein to a white protein. We're breaking that protein structure down. And then the SDS is a detergent. It's like a soap. It's got a charged part and a hydrophobic part. Basically, what that does is it is going to help to further break down the structures, but it's also going to coat the protein. Kind of like if we kind of had this ribbon again, and we decided to put it in some flour, you could imagine the flour would kind of get in all these ridges and basically coat the ribbon, with a powdery coating. SDS is going to coat the protein. It's going to interact because it's going to take its hydrophobic tail and interact with areas in the protein. The charged heads are going to remain, outside, and it's going to help give our proteins a general negative charge. So now that we've got our secondary, our tertiary, our quaternary, and our secondary structure broken up, we essentially have long chains of proteins. We've made them negative by coating them with this SDS, which is going to allow them to then all be negatively charged regardless of their actual sequence. Once we have all of those things taken care of, we can then load them into a gel, turn on the electrical field, and separate the proteins by their size through the porous matrix, through the porous gel molecular sieve. And the proteins are going to migrate in through the polyacrylamide gel in a predictable manner. So much like DNA electrophoresis, larger molecules, it's going to be harder for larger molecules to fit through all of the pores in the gel. Smaller molecules are going to fit through more easily. So smaller molecules will be found near the bottom of the gel, larger molecules near the top. One caveat is many proteins are going to be clear and colorless in solution, and it's even proteins like GFP that are green in solution. If you break down their secondary structure, their tertiary structure, they're no longer going to have a color. And so we need a way to visualize our proteins. Now with this particular experiment, our proteins have been pre-stained. And that means, we extracted the proteins and we chemically cross-linked them to blue dyes. And that means that we can actually see the proteins in the gel without any sort of counterstaining. And that's nice because it allows you to run the experiment more quickly and get results. However, to enhance the results, we have developed a new stain called Flash Blue Protein Stain. And this is going to be rolled out in our kits, in our protein experiments very soon. It's different than the Intastain you might have experienced previously with protein kits. This is actually going to it will come to you as a powder. You add vinegar and ethanol to this powder to make a liquid. And then you're going to stain the gel in this liquid. There's a couple things I really love about this stain. The Instastain cards were great, but you did need to use acetic acid, concentrated acetic acid, and methanol to be able to be able to stain the gels. And acetic acid is fine when it's diluted. It's basically just vinegar. The bigger problem is the methanol. And the methanol can be a problem in class. This uses ethanol, which is easier to use, and you don't have to worry about it with your students as much. I like to use this dye again because of the ethanol. You are going to cover. It's really fast. It's faster than the InstaStain paper. You're basically going to cover your gel with a little bit of the dye, microwave it for 10 seconds, sit it in the stain for 15 minutes, and then just destain in a solution again of ethanol and vinegar. And I have that here. I'm not going to bother to show that bottle. It's just a clear bottle full of clear liquid. But it's fast, and you can destain the gel fast. You're using fewer hazardous chemicals, and you can get great results. You visualize it on a white light box, and we can see the results. And so actually, we are at the point now where we are going to look at the results of our experiment, which is always the fun part. And it's good because we only have 10 minutes left, which is crazy. These workshops always go so fast. So I'm going to turn this off. This gel's been running for about 30 minutes or so. I don't remember the exact runtime in the manual, but you're going to see that, what we can get even with a shorter period of time that is recommended. I also have a second gel that I have pre-stained so that we can compare it. But let's take this out. I'm going to move my True Blue. Let me get the gel out first, and then we'll adjust. You know what, even without, you can see the foam is from the SDS. Again, that's sodium dodecyl sulfate. It's a detergent. We find it in our soaps. We find it in toothpaste. And it basically allows us to solubilize molecules. And you can see, even here, without a white light source, without even staining the gels, the proteins, you can see we get really great resolution. You can get really great resolution between the different bands and really see them clearly. The question is, can the new Flash Blue stain be purchased on its own? It will be launched on the website in the coming days. I don't know exactly when, but it will be a standalone item. I'm sorry to hear you've had terrible luck with the cards and destaining. They can be tricky, but the new Flash Blue stain, I think, will make your life so much easier. And so, Maria, if you want to maybe, or you know what I would do, Rachel, is put that in if you haven't filled out the form yet, or even if you have, you can put that in the form and we'll reach out to you, or you can reach out to us on Twitter. I know we tweet with one another, and so we can let you know when that launches. But here are the pre-stained proteins. Our protein ladder. Let me show you the results, and we'll compare this next to a gel that has been pre-stained. So you can use a white light box if you have it for this. I happen to have the True Blue 2 here at home. Okay, we're also going to be announcing it in an email too. So then you will definitely know if you are signed up for our email list. Okay, so here is our gel. You're going to want to crack this out of the cassette generally if you're doing this. I did not crack it out of the cassette just for time. But basically what you can see here is that we have our ladder, and then we have our 3 unknowns of different sizes. What you would do is you would take these unknowns and you would determine you would use the known bands here in our standard ladder. So these are proteins of known size, and you would calculate the protein molecular weight using a standard curve. That is a pretty long technique. You can take a picture of this gel. I like to just take a cell phone picture and print it out. You'd measure the distance that each of these traveled, plot it on a semi-log paper, and then you would calculate using these distances traveled. And I actually have a really nice video on our YouTube channel. You can find it. It's called Calculating Protein Molecular Weight, or Calculating Molecular Weight Using a Standard Curve. It's for DNA. Making a standard curve is going to be the same for protein. So definitely check that video out. But basically, after you were to do that exercise, you would find that these 3 proteins have 3 different molecular weights. And we have, our protein standard marker here to compare them to. So this is, again, this is the pre-stained proteins, but I took one of these gels last night, I ran it, and then I stained it with the Protein Flash Blue. And that is going to be this gel here. And you can see that it just enhances the blue color. It makes the bands a little bit more sharp than being pre-stained. And we get these dark blue bands on a light blue background. And it's simple, it's fast to use, and I feel like it's using less reagents that can be, fewer reagents than that you don't want your students to get into trouble with. Again, so, you're using the ethanol instead of the methanol, which is going to reduce that hazard there. All right. So I hope you enjoyed doing this experiment with me today and learning more about protein electrophoresis. Oh, one more thing I want to go back here. I forgot to mention. So we measure protein molecular weight with a unit called a Dalton, which is, again, a measure of protein molecular weight. 1 Dalton is defined as the mass of a hydrogen atom. And so the hydrogen atom, a kilodalton, which is what I have here, a kilodalton is going to be 1,000 daltons. So what is the scientific reason for smearing? So what I would say is that some of the smearing is going to happen if your proteins are not fully denatured. They're going to end up running faster or slower. If they're a little bit degraded or something's a little bit sticky in the wells. Sometimes SDS can be a little sticky too, which is going to get some of that. For here, I would just say, it's probably just the nature of proteins, the way they're denatured. It's really rough on the proteins. It's going to be not only, it's heat, it's detergent, and it's SDS. And basically, you're blowing these proteins up. For the pre-stained proteins especially, some of it is going to be excess dye from the pre-staining. But any dye from the loading, you're going to see in a band down here because it's small, it's very small, and it's going to run through really quickly. It's really just that everything is a little bit sticky, and we just are able to see that because it's sticky. If you see anything stuck up in the wells here, it generally would mean that your protein isn't denatured well enough, and it's just so big and gunky it gets stuck there. You're always going to see a little bit of background with protein electrophoresis. It's just kind of the nature of the chemical interactions. And especially if you're using a very crude extract, I know you mentioned 403. The protein samples that you're running haven't been super purified. So there is going to be stuff in there that just is very, very kind of gunky. And then again, that's just because cells are complex. They've got a lot of stuff in them. And even with the column purification, there's still going to be other stuff around that interacts with the proteins. Okay, so in summary, proteins are built from amino acids. Each amino acid has characteristics to it that is going to affect the way it interacts with its neighbors in the protein. And the amino acid sequence is going to determine the 3D structure of the protein, how it interacts with other proteins, and how it functions. Higher order structure does make proteins difficult to separate. I'm reading and I realize I have a little bit of a grammatical error in this slide. But the higher order structure makes proteins difficult to separate by electrophoresis. Again, because proteins can be chemically linked to one another. They can be, in very tight secondary, tertiary, or quaternary structures. And they just don't want to go into the gel in the same way that DNA does because they have different charges based on their side chains. And so what we need to do is denature the proteins, and then we can separate them by size using Sds Page or polyacrylamide gel electrophoresis. So with that, we are Edvotek. If you want to request this presentation, please be sure to fill out the form that Maria has put into the chat box. We are going to be posting the presentation and the slides in the next few days. We, again, fill out that form. If you have any other questions, let me know now, and I will be happy to answer them. But here we have our social media. Oh, I forgot to put our TikTok up on here. We are If you need to get in touch with us, we want to hear your questions. We want to hear your concerns. Feel free to reach out to us by phone, by email, or by social media. You are here watching this on our YouTube channel. We have a lot of really great content here. Be sure to check it out. We try to post it frequently. You can see my gel is curling as it dries out. You do want to take pictures of these gels quickly because they dry out and they kind of shrivel. I perhaps have found one that I've left out for weeks and it kind of becomes like a little shrinky dink, which is kind of fun. Again, but this is basically like a plastic polymer. It's hydrated with water, and as that water evaporates, it kind of shrinks. We also have social media. We're on Twitter, we're on Facebook, we're on Instagram, and we've started a TikTok channel. We're Edvotek over there too, where we have a lot of really fun TikToks that talk about different experiments. Thank you for linking. Yeah, definitely take a look at the TikTok. We try to put up a new TikTok every Friday. So subscribe there. Subscribe here to YouTube to be alerted of all of our new videos. Make sure you hit like on this video too. I'm always the worst YouTuber because I never tell people to like our videos. Definitely like our videos. It'll make sure you can really see our content. Thank you so much for your time today. I really enjoy being able to discuss these biotech techniques with you. And I look forward to seeing you next month for another biotech live stream. Have a great afternoon or morning, depending on where you are, and stay safe. We'll talk soon. Have a great day.